Hello everybody. So we'll be talking about cell membranes today, and just a disclaimer, if my audio starts being really bad, I am so sorry. But yeah, y'all have your discussion on this, so we thought maybe it would be a good idea to do a video about cell membranes, talking about I guess more context on them and more information that might help you throughout your studying. There's also a practice question at the very end of this video. If you want to take a look at that now, you can totally do that. But yeah, we'll just get into this concept. Before we go into more detail about cell membranes, I do want to talk about the second law of thermodynamics. If we were to very much oversimplify this law, the second law states that in an isolated system, over time, entropy increases. Entropy, you might have heard this word from your chemistry or physics classes, but entropy, also modeled by the letter S, is our measurement of disorder. So we can take ice melting into water as an example. So this is my makeshift ice cube, ice, and this is my makeshift water. It's kind of weird. I can make that look more flowy, but it's all good. This is our water. So if we look at ice, right, ice is highly organized. It is highly structured. These water molecules, H2O molecules, are in very compact orientations, and there is less freedom of movement in this structure. So ice being very organized is going to have a lower entropy. When we melt ice into water, water molecules are able to move more freely compared to their previous conformation. Water molecules will hydrogen bond and rehydrogen bond to themselves, resulting in that finicky, flowy characteristic that they have. Water molecules are thus more disordered. They have less organization. They are less compact in the way that ice is, compared to ice, I should say. And so water is going to have a higher entropy, a higher amount of disorder, a higher amount of disorganization. Water naturally has a higher amount of disorganization compared to ice. So you can think about entropy when you take a sip of water. With living organisms, we have a way of combating the second law by decreasing our own entropy. This is at the expense of increasing entropy within the environment. So whatever we decrease in entropy, right, there has to be an increase of entropy somewhere else. So if we're decreasing the entropy within ourselves, living organisms, I should say, we are increasing entropy of our environment. This allows us to maintain an internal composition in our cells that's different from their surroundings. And so we need to be able to control a barrier that lets us maintain that separate environment. That barrier being our membrane. So we can get into the composition of membranes now. You can also call cell membranes bilayers. These are made up of lipids. Just a general slide about lipids. These are macromolecules that are nonpolar and hydrophobic. They are filled with hydrocarbons. By hydrocarbons, I mean those CH bonds. These guys are important for energy storage, important for structure, membranes, and pigments and chemical signaling, like hormones. Examples of lipids are testosterone and cholesterol. Cholesterol is pretty important for membranes as well. This helps in fluidity of membranes. So at the very end of this slide, I do have a question that relates to cholesterol. So knowing this guy is responsible for or contributes to fluidity of cell membranes. That's important, at least for the question at the end of the slide. Lipids also include ester linkages, the bonds between them, and they're formed via condensation reactions. So condensation reactions and dehydration synthesis, we refer to these two names as the same thing. In your OChem classes, if you ever decide to take organic chemistry, you'll find that condensation and dehydration synthesis are different in the way that they happen, but for the purposes of this class, you can associate condensation reactions and dehydration synthesis as the same thing. You may look at lipids in different ways in your midterm lecture or discussion. These are some of the ways you might see them. In this first structure here, we can see those individual CH bonds that make such a large nonpolar character within lipids, and this is what helps characterize lipids as what they are. Of course, we also have very, very small amount of OH groups. We have one OH group here. You may also see phospholipids that look like these two. These individual points, right, are carbon atoms, and we imply hydrogens at each point. Same here. These are where the hydrogens are. There are lipids that are saturated. These are your saturated fatty acids, saturated in hydrogens. You also have lipids that are unsaturated, and they usually have a double bond. These are unsaturated with hydrogens. This example here is not to fool you or anything. This is cholesterol. This is also a lipid. This is cholesterol drawn out. And even though we have hexagons and pentagons, these are all still CH bonds, right? This is still very highly nonpolar, as you can see. So this is also an example of a lipid that you might see in lecture or discussion. We can get just more detail about saturated and unsaturated fatty acids. Your saturated fatty acids, these are going to be hydrocarbons with no double bond. So this is your saturated one. You can think of these guys being saturated in that there is an abundance of hydrogens. Saturated in that they are saturated in hydrogens. In contrast, we have unsaturated fatty acids. These are hydrocarbons with a double bond, and this results in kinks in the chain. This one here is your unsaturated fatty acid. And because of that double bond, we have less hydrogens present here, are less saturated with hydrogens, hence our unsaturated fatty acid. That's one good way of looking at these two different structures. And our double bond results in kinks. I didn't really draw the kink out, but we'll see that in the next slide. I do have a question for you. So which do you think, just based on the structure that we looked at, which do you think have a lower melting point? Saturated fatty acids or unsaturated fatty acids? So feel free to pause the video and to think about this on your own. We can reconvene in a little bit. Alrighty, welcome back. So in terms of which would have a lower melting point, we're looking at unsaturated fatty acids. Why is that? Well, you can thank the kink in the unsaturated fatty acid chain for that. So unsaturated fatty acids, under normal temperatures, they're going to be in a liquid form. Saturated fatty acids, under normal temperatures, are going to be in a solid form. It takes much more heat to melt saturated fats. So this is why your unsaturated fatty acids are going to be lower in melting point. You can thank the kink in the chain for that, the double bond. Okay, so now we can get into the composition of cell membranes. So your bilayer or your cell membranes, they're composed of phospholipids, and these are a type of lipids with phosphate groups in them. You have your phosphate, and you have your glycerol, and you also have your R group, and these guys make up the polar head. I wrote down choline here because that's one example of an R group that you might find in a phospholipid. Choline is not the only one, hence the R in green. And then you're also going to have non-polar fatty acid tails. I didn't write the correct number of CH bonds here, but this is just to give you an idea. We have our polar head and our non-polar tail, and you can see the kink in the chain here because of the unsaturated fatty acid, the double bond within unsaturated fatty acids. Cell membranes are going to have a composition that looks a lot like this. And as you can see, this is a very organized, very structured way of surrounding the cell. Very organized, very structured, right? So how does this happen? Membranes form spontaneously, and we can thank hydrophobic effect for that. Hydrophobic effect is an entropic force. So we talked about entropy at the very beginning of this video, right? This is where it's coming in now. Hydrophobic effect is an entropic force, and it explains the tendency of nonpolar molecules in aqueous environments to aggregate together and or displace water molecules away from them. So we saw this in protein folding as well. Your tendency for nonpolar amino acids to be at the center of your protein and your polar amino acids to be on the outside of the protein is also thanks to hydrophobic effect. And this is just the tendency for these polar amino acids to rather hydrogen bond to themselves than to the non-polar amino acids at the center. And so these polar amino acids act as a shield in a way, and they can interact with other solutes within the cell. The non-polar amino acids are going to be at the center, protected from other polar substances. If we think of hydrophobic effect in the context of cellular membranes, we're looking at something similar. We're looking at the orientation of water molecules around nonpolar solutes, that being our lipids. So in the formation of our membrane, or at least before the membrane forms, we have something that looks like this. These long ovals being our lipids. And then I guess in yellow is going to be our water. So like I said earlier, right, water molecules have a tendency to rather hydrogen bond to themselves than they do the nonpolar solute. And so they want to maximize the number of hydrogen bonds they make with each other. As a result, they straddle the solute, the nonpolar solute, and quote unquote form a cage, a water cage. You can see the water cage is kind of surrounding the nonpolar molecules. Now, saying water cage is actually not a correct way of describing membrane formation necessarily, but I'm saying this in the context of it being a good analogy for what this kind of looks like. Water cage, again, is not a correct way of saying this, but let's just say, I guess in more detail, your water molecules because they want to maximize the number of hydrogen bonds they make, are going to be more organized in certain places. And so this is why you have water molecules kind of looking like a cage around the nonpolar solutes. This orientation that we have here is not favorable. So if we were to think about this in an entropy perspective, if we have water molecules that want to maximize their hydrogen bonds, by the way, hydrogen bonds are are stronger than dispersion forces. Dispersion forces are interactions between nonpolar solutes, and so it's harder to break those hydrogen bonds. But they want to maximize those bonds, right? Those polar interactions, those dipole-dipole interactions. And so they're going to be more organized in a certain place. What do you think the water molecules then, if they're organized in a certain place, what do you think the entropy would be for the water molecules if they were in a conformation like this? So they would be higher in entropy. No, sorry. They'd be lower in entropy, sorry. So why is that? Water is in a more organized state here, forming those quote-unquote cages around nonpolar solutes. Now, what we decrease in entropy in one place, we have to increase entropy in another. So the entropy of our lipid is going to increase. This is not favorable. This is not a stable conformation. H2O, water, is naturally higher in entropy because of its finicky, flowy characteristics. Its want to hydrogen bond and rehydrogen bond to itself and to be free-flowing. We do not want water to be lower in entropy because that's not its natural tendency. So water and the nonpolar solutes are going to change their conformation spontaneously so that nonpolar molecules cluster around sort of like this, and your polar molecules get away from the nonpolar ones. And so when we have a conformation like this, water molecules are able to hydrogen bond with themselves and other polar groups. These yellow dots would be other water molecules. Same thing with down here. They're in a less ordered state. They're in a more free-flowing, free-moving state, as water should be, right? Your nonpolar molecules are going to be compact and close together. They're going to be in a more organized shape compared to what they were before. So what do you think the entropy of the water will be if it is in this state instead of the quote-unquote water cage? Well, your entropy of the water is going to be higher. And your entropy of the lipid is going to be lower. We want the entropy of water to be high. We want water to be able to be free-flowing, free-moving, to be able to hydrogen bond and rehydrogen bond to itself, to have that finicky, flowy characteristic that it has, as we talked about with ice melting into water. Our lipid, as an expense, becomes more organized in that they're stationed or oriented in a manner that is less disordered. So you can see there's more organization for the lipid, hence the lower entropy. This conformation is a more stable and favorable conformation for our membrane, and over time it will look like something like this, as we've seen in this slide. This is how our membranes form spontaneously and how hydrophobic effect contributes to the formation of cell membranes. Okay, so now we can get into function. Your bilayer or your semi-cell membranes are semi-permeable, meaning they are highly specific and highly controlled in the way that they manage the import and export of substances in cells. They allow certain things through. They also don't allow certain things through. So what can pass the bilayer? Well, for sure, hydrophobic nonpolar molecules can. Things like CO2, oxygen, testosterone. You can also see small uncharged polar molecules like water and glycerol be able to enter or pass through the membrane. Water can pass through via aquaporins in the membrane. So they can go through the membrane that way. What can't pass the bilayer, though? So on its own anyway. We're talking about large uncharged polar molecules like glucose. Glucose cannot move through the membrane on its own. It's going to need help from proteins. Same with ions. Anything charged is going to need assistance from proteins in order to pass through the membrane. And sometimes energy as well. So I also want to talk about osmolarity and tonicity. Your cells are, it's not just your cells, my cells too, all of our cells are majority made up of water. Our cells are, the inside of our cells and our cells themselves are in a constant aqueous environment, uncharged polar aqueous environment. So if we were to draw our cell, right? This is our cell. Cells tend to be hypertonic, and by hypertonic I mean there is a higher concentration of solutes and a lower concentration of water inside the cell compared to outside. So we have a higher concentration of water outside the cell, lower concentration of water inside. We have a higher concentration of ions. This is because of the counter ions within macromolecules. So if we have a lower concentration of water inside the cell and a higher concentration of water outside, by process of diffusion, and diffusion, or I guess osmosis since we're dealing with water, deals with the movement of solutes, substances I should say, from high concentration to low concentration. Water molecules are going to want to go inside the cell for this reason, from a high concentration to low concentration. What happens if we have too much water entering the cell? You can think of a blood cell too. As a result, our cell is going to swell up and burst with water. And we don't want that. We don't want our cells to burst. We don't want them to swell up, especially because of water. Nothing wrong with water or anything, but we don't want our cell to swell up. We don't want it to burst. And so there has to be certain ways in order to combat the problem of osmotic pressure. Problem of osmotic pressure. So certain organisms deal with this in certain ways. With prokaryotes and also plants as well, you have the addition of a cell wall. So my professor, when they taught this to me, they used a bag in a box analogy. So what you have is a cell and then a wall that surrounds the cell. Your bag here and then your box here. This allows the cell to maintain a hypertonic environment in which you have a higher concentration of solutes, lower concentration of water. It keeps more water from entering the cell because of the cell wall, right, that blocks its way. The cell wall also has certain characteristics that allow nutrients in and out, therefore, it being a very highly controlled, highly specific form of protection against osmotic pressure. So prokaryotes have that cell wall, so do plants. They also have a cell wall. For eukaryotes, though, for organisms like us without cell walls, we have to fight osmotic pressure differently, and we do this through endocytosis. Oh gosh, you guys didn't see that. Okay, I should more like specify exocytosis, but let's go over endocytosis. This is the process of taking in matter into the cell by creating a cavity in the membrane. This forms a vacuole. So if I were to draw this is a piece of food. This is our cell. Our cells have the ability to engulf the food. Changing the color here. To engulf the food. And this is a benefit because it allows us to privatize our food. With certain prokaryotes, certain organisms who do not have endocytosis, competition for food is great. You are more likely to have your food stolen from you, especially if you can't privatize it. With endocytosis, we're able to take in nutrients and keep it into our cell, protect it so that we can break it down and use it as a form of energy, and keep other organisms from stealing it from us. Now, this is not to say that there's no competition of food for eukaryotes. There will always be competition for food, but endocytosis allows us to privatize the food that we absorb, and it also allows us, eukaryotic cells, I should say, to engulf bigger things. What does this have to do with osmotic pressure? I'm glad you asked. When we're taking in food from the environment, as you can see, we're not just taking in the food, we're taking in bits of our environment with us, aka water. We're taking in water. And so we take in a part of our environment with us, but in exocytosis, which is a process of taking out matter into the cell by releasing this cavity from the membrane, we do the opposite of this process, right? This is our cell. This is our piece of food, waste that we want to get rid of. We form a cavity. I'm going to take more time drawing this than I am explaining it, but we form a cavity that allows the food to exit. But it's not food, the waste to exit, but it's not just the waste too, right? It's water as well exiting. So with endocytosis, exocytosis, we are able to take in a part of our environment, but we're also able to take out of it. So we can maintain our hypertonic solution within our cells by constantly taking out the water that enters. So we do this through exocytosis, right? while water is able to go in because we do not have a cell wall, we are able to take it out in some way. And so we can still maintain hypertonicity within our cells. And this is the way that we fight osmotic pressure. Okay, you have also probably seen this in your discussion or lecture, but it's important to look at a membrane or cell membranes as like a mosaic of things, and it being very fluid and able to move. So a lot of things contribute to the way that cell membranes work, the way that they're highly controlled and highly specific, of them being our proteins, our transmembrane proteins. We have protein channels, we have carrier proteins, we also have peripheral proteins, and then these are our phospholipids and our cholesterol. We have glycoproteins. A lot of these are helpful for cell signaling. Also, a lot of these are helpful for the import and export of solutes. Cholesterol, again, I want to emphasize, this is your nonpolar substance, and so it's going to be attached to the nonpolar parts of the phospholipid. This is very important for the fluidity of the membrane. I kind of foreshadowed that at the end of this video, I do have a practice, and part of it relates to cholesterol. So knowing that this contributes to the fluidity of the membrane is really important, at least for that question later on. Same with your unsaturated fatty acids. Those are very important for fluidity as well. Okay, so what is a membrane without its ability to transport things, right? So we're going to talk about membrane transport, the first one being passive transport. There are two ways of that we learned about in passive transport, that being passive diffusion, or you can just call this diffusion. The other being facilitated diffusion. But we should also define what passive transport is too, right? So passive transport involves moving substances through the cell without the use of energy. This is usually in concentrations from high outside the cell to low inside the cell. And we saw this before, right, with the osmosis slide. With passive diffusion and facilitated diffusion, let's just say diffusion in general, we're talking about solutes moving from a high concentration to low concentration. This is down concentration gradient. So for passive diffusion, right, if we were to draw our membrane, passive diffusion is diffusion without the assistance of proteins. So our nonpolar solutes, substances like oxygen, CO2, testosterone, things like that, are able to move down their concentration gradients through the membrane via diffusion. So I can take O2, I can put O2 out here. Let's say that this part is the outside of the cell and this is the inside of the cell. Oxygen can move down its concentration gradient through the membrane, and you can see there's no assistance of proteins needed here. For facilitated diffusion, we're still moving down the concentration gradient, but now we need help from a protein. So we can take a substance like glycerol, for example. Once I draw this membrane, I put an extra thing there. Let's take glycerol, for example. There's a higher concentration of glycerol outside the membrane than inside the membrane. We're going to need a channel protein to be able to move the glycerol through the membrane. down the concentration gradient. So this in blue is your channel protein. This is facilitated diffusion, facilitated being like also known as, with the help of, and that being proteins. So these are your two different kinds of passive transport. You can also see that we have active transport involved in membranes as well. Active transport is the opposite. We're talking about moving substances through the cell membrane with an energy input. Usually with active transport, substances are going against the concentration gradient. We defined going down the concentration gradient as naturally how diffusion works. So going from high to low concentration of solutes. Going against the concentration gradient means we're going from low to high. Low concentration outside of the cell, high concentration inside. This is our against I'm stuttering so much, against concentration gradient. So if we were to draw the membrane again, this is our membrane. We need the help of carrier proteins, transmembrane proteins, in order to move solutes like glucose, for example, against the concentration gradient. So let's take in glucose. There's a higher concentration of glucose inside the cell than outside. But we still want more glucose, right? Because glucose is a form of food for us that our cells need, and so we're going to want glucose anyway. In order for glucose to go against its concentration gradient, we need to use energy in the form of ATP. This allows glucose to be pumped into the cell. So with active transport, we need a pump. With passive transport, we need a channel. These are two different things. So I do want to emphasize that. And with active transport, we are using energy. There are different types of proteins that you might see. We have symports, for example. So take this blue thing as our protein. Symports allow substrates to go into the cell in the same direction. Antiports, let's put symport here. Antiports, they allow substances to go in different directions, two different substances to go in two different directions. A good example of this is the sodium-potassium pump, which we'll actually look at in the next slide. I'll briefly explain it, although it's a very simple overview, I guess. But antiports, these are also used for the transport of ions. So when we're talking about the transport of ions, especially against their concentration gradient, we do active transport. We need that energy in order for that to happen. So if we look at sodium-potassium pump, for example, our sodium-potassium pump, this is our carrier protein, and it kind of looks like this. All those divots in the protein are places where ions like sodium, let's get our sodium here, can bind. When sodium binds to the protein changes shape. Let me put that there. The protein changes shape, making something like this. That's not the greatest look, but our sodium ions are kind of over here. This allows the release of other ions, that being potassium, out. And this is with the help of energy as well. We need energy in order to do this. The antiport can go the other way too, right? We can take in these potassium ions, change the shape of the protein, and allow sodium ions to be released instead. So this is your sodium-potassium pump, and this requires energy in order to work. We see this in our membranes a lot. This is like a classic example of how active transport works in our cells. So this would be a good thing to definitely review as well for your midterms, finals, or just for your own study needs. As far as content, we're actually done here. This is all I really wanted to show you, so we can get into that practice question that I alluded to. In this question, we're talking about cholesterol. Like I said before, we're also talking about saturated and unsaturated fatty acids. So I kind of mentioned earlier that cholesterol is very important for the fluidity of a membrane. Same with unsaturated and saturated fatty acids. They also contribute to the fluidity of membranes. So these are some good things to know before we approach the problem. If we look at the question, it says that goldfish are capable of spending a part of their life under extremely cold temperatures and a part of their life under normal temperatures. They can also hibernate. What change in their cell membranes might occur as their environment shifts from normal to colder temperatures? So I'd like you to think this through. I'd like you to think about what the question is trying to ask you, what concepts the question is trying to test you on. A lot of these answers have to do with cholesterol and some fatty acid, whether unsaturated or saturated. I want you to think about your knowledge of unsaturated and saturated fatty acids and to deduce what the question is asking you. So I'm going to pause for a little bit. I want you to pause the video as well. Work this out on your own, think about it, and then we can reconvene in a little bit. Okay, welcome back. So let's briefly go over the information that we learned throughout this video and also information that you've learned in your discussion and lecture. So a lot of these answers have to do with unsaturated and saturated fatty acids. Let's go over just a small overview of what those are. Unsaturated fatty acids, they have a double bond. We also found out that these guys have a lower melting point. Under normal temperatures, as I've said, your unsaturated fatty acids are going to be in a liquid state more often. Now, your saturated fatty acids, there are no double bonds. You have a higher melting point. Under normal temperatures, your saturated fatty acids are going to be in a solid state. So it takes much more heat to melt saturated fats in their solid state. Okay, so let's look at unsaturated fatty acids again, right? Under normal temperatures, they'd be in a liquid state. They'd be in a more free-flowing fluid state. With saturated fatty acids, they'll be in a less fluid state. If we're going from a normal temperature, maybe 25 degrees Celsius, right, to colder temperatures, to extremely cold temperatures even, do we want more fluidity in our membrane or do we want less fluidity in our membrane? We want more fluidity because we still want to be able to control imports and exports in the cell. We still want to take in nutrients. We still want to do everything that membranes, as we've learned, do. under colder temperatures, we don't want production of energy or nutrient uptake to slow down. We still want to be able to do that. We want to be able to survive. In order to do that, we have to take in nutrients in some way. And so we want our membrane to be more fluid. That means we should increase the production of unsaturated fatty acids. I also want to mention with unsaturated fatty acids, if we have kinks in the chain, right, that creates holes in the membrane that's able for it to be more fluid. But if our membrane has way too many unsaturated fatty acids, this can also be a problem because of those holes. In the case of colder temperatures, having more unsaturated fatty acids is optimal. We want to increase that fluidity. I also mentioned that cholesterol contributes to fluidity of membranes. So would we want to increase the production of cholesterol or decrease it? We would want to increase it. So the answer to this question is A. The goldfish's cells would increase production of cholesterol and increase production of unsaturated fatty acids. This is to build fluidity under cold temperatures. So I hope this question was good practice for maybe something you might see on your midterms or finals. It is good to look at how your professor may quiz you when it comes to cell membranes. There could be a variety of ways. I hope this video has helped throughout your studying. I believe in y'all. Y'all are going to do great on your exams. I wish you best of luck. Thank you so much for watching. Until next time, bye y'all.